Good morning, everyone. I hope you had a good weekend. Let's start with a little bit of recap of the blood type that we learned last class. So let's just do like a warm-up question. Let's say we want to take blood from a type A person, and then we're going to give it to a type B person. Let's revisit whether this is safe or not safe. So this is our donor right here on the left side, and this is the donor, and this is the recipient, recipient. And we are going to be performing red cell transfusion. And so we need to look at the antigens that are present on the donor's red cell. In this case, the person has type A blood, which means on the surface of the RBC, there will be type A antigen. Okay, these are just antigens are proteins, carbohydrates sometimes, and lipids that are found on the surface of the cell. And we have the A antigen right here in the donor cells. Now let's take a look at the recipient. In the plasma of the recipient, what kind of antibodies will they have? Well, this is a type B person. So as a general rule, you will make antibodies for things you don't have in your body. And if you're type B, then you don't have type A antigen, and so there will be anti-A antibodies. So if we go ahead with the blood transfusion, you will see that the anti-A antibodies will now attack the incoming red cells. Okay, so this would be the red cells coming in, and then the antibody will basically attach. I got to restart the program. It's not working. Just give me 1 second. Then the antibody, since it's anti-A antibody, I should probably write down for you. Anti-A antibody. It's gonna recognize anything that has A on it. It's gonna tag it. And basically, it will cause a bunch of these red cells, right, to clump together, right, to clump together. There might be another red cell here, and this clumping is called agglutination. Okay, agglutination, clumping, and that's gonna lead to hemolysis. Right? Hemolysis is basically like the red cells all breaking apart. Okay, it burst. Okay, they all are gonna leak all over the place and they basically gonna die. Okay, so here we say there's a cross reaction between the recipient antibodies and the donor's red blood cell. Missing an I here. Okay. And the cross reaction will lead to agglutination, all these things clumping together, and finally it will lead to hemolysis. Right? So all the transfused red cells will basically burst, and that's going to make the situation worse, right? Remember, the reason a person needs blood transfusions is because they don't have enough red cell. So if all the new red cells that are coming in is going to get destroyed, then number one, you're not fixing the problem. Number two, you might make the situation worse because now you have all these red cells dying, and they're going to be leaking hemoglobin all over the place. The liver is going to have to work extra hard. Spleen have to work extra hard to cope with all these red cells that are dying, and it's just going to make the situation worse. So in this case, this is a incompatible blood transfusion. Okay, I'm gonna go back to the PowerPoint for just a moment, and then we have a lot of notes to take today, guys. So warm up your fingers. All right, so this is what I'm trying to show you, right? Uh, Now in the notes, I draw the red cells with the A and the B on it because I think it's easier for you to visualize. But a lot of textbook, they just use shapes to represent the A's and the B's. So try to follow along here. We have a type A donor here, and the A antigen is represented by these spheres, okay, these circles. And so that's A antigen. We'll say antigen A, okay, antigen A. Remember, antigens are on the surface of a cell, whereas the antibodies are in the plasma. So this is giving type A blood to type A blood. Obviously, that's going to be okay. You can always transfuse blood of the same kind to another person. But let's take a closer look at why this is okay. If the person is type A blood, then they have anti-B antibodies, okay, in the plasma, right? And you can see the the ends of the antibodies, the shape is for triangles, right? It's not going to recognize the circle. Right? If I have to put letters on, these would be A's, and then this would be B. Right? The B and the A are not going to recognize each other, and so the antibodies of the recipient, the person receiving the blood, will not be attacking, will not be cross-reacting with the incoming red cells, and it's going to be fine. Right? So if you take the donor's red cells and mix it with the plasma of the recipient in like a petri dish, you can do this outside of the body. You will see there's no clumping. Okay, you just see the blood kind of sit there. It's not going to be uh, aggregating together. It's not going to be clumping. So no agglutination. In this case, we are taking a type A blood and giving it to a type B person, I believe. And what you can see is the donor will have the spheres again, representing the A antigen. And this is a type B recipient, which means they will have anti-A antibodies. And you can see the ands here, it will recognize the round shape as opposed to previously, the anti-A recognizes like triangles, right? So these are anti-A. And if you go ahead with transfusion, as I showed you in the example at the beginning, these will bind to each other. It will cross-react, and then it will cause clumping, agglutination. You can see it um, under the, you don't even have to put this under the microscope. Like you literally just do it in a dish, and then you will see that they form these little clumping bits. I have a video on Luminate that shows you how it works. Here, let me just quickly show you where it is. Because it's sometimes it's hard to visualize these things, right? So again, go to your courses. Um, if you move to the lab, okay, I see that not a lot of you have seen this yet, so please spend some time looking at it. The lab videos. Okay. There is the blood typing simulation. You can take a look at that, and then you will be able to see how it actually clumps in a dish. Right? And that's something that they do before blood transfusion. Sometimes they'll match to see if the bloods are compatible. And this is also a good video, blood typing explanation. If you have trouble understanding some of the stuff I am explaining to you. Please take a look at this. It's like a model with the red cells and then with the antibodies and the antigen, and it's all color coded. Just take a moment and watch that. It will definitely help you understand more. I'll put it in the chat box as well if you need it. But everything is under the lab video folder um, of lab 1. All right. So that's the clumping all over the place. That's it. Any question about ABO blood group before we move on to talk about RH? Okay. When I asked you what are your blood types, and some of you tell me you're A positive, B positive, O negative, right? The positive and the negative is actually talking about the RH blood group. Okay, the RH blood group is named after these monkeys. They are called rhesus monkeys, and it was first discovered in them, and humans have it too. So we name them RH blood group. So I'm gonna give you a very long note, okay? It's probably gonna take us like, half an hour, 45 minutes to write the whole thing. Uh, so, get some papers, get some colors, and be prepared to write a lot. Okay. Let's start with what RH antigens are. Okay, RH blood group. Okay, so again, the RH stands for rhesus. So I'm just gonna give you some bullet points here. There are two alleles. Two alleles for the RH blood group. As opposed to how many alleles for the ABO blood group? How many alleles do we have for the ABO blood group? 3 is correct, right? IA, IB, and little I. For RH blood group, there are only two alleles, and they are positive and negative. They don't have any special letters associated with it. Okay. A person can be positive, positive, or positive, negative. Both of these genotypes will give RH positive blood type. Negative, negative will give RH negative blood type. Okay. So what does it mean? Well, if a person has the positive allele, okay, the positive allele allows the synthesis of RH antigen on RBC. Remember, these are genes. Genes has the instruction to make proteins. In this case, you're making the RH protein, and you're going to put it onto the red cell. So the only way to make RAH is if you have the positive allele. If you have the negative allele, you cannot make it, right? And you don't need to have two copies of the gene to make the RH. One copy is enough. Right? It's not about how much RH you have on the surface of a cell. It's all about whether you have it or not. If you have the RH antigens, then you are RH positive. If you don't have it, then you are RH negative. And in order to have RH antigens, you need to have at least one copy of the positive allele? Just gonna pause there for a second, let you think about it. We good? You do not get it? Okay. Do you have a more specific question? Which aspect do you not get? Do you like you don't understand the difference between Rh positive and negative, or you don't understand why the alleles would result in the blood type? Why the allele gives blood type? Okay. So again, if this is the chromosome, okay? This is the chromosome, and then the person will have the Rh gene, okay? So let's say they have positive and positive here. This is the name of the allele, okay? Positive allele and positive allele. What does it mean? It means this gene will allow the cell to make Rh. So if you look at the RBC, right, because of the positive allele, they now can put Rh on it. And because the cell has Rh, the person will be Rh positive. Okay. But then, like I said, you don't necessarily need to have two copies of the Rh positive gene. You can just have one copy that's positive and the other is negative. So this one will not make Rh for you, but it doesn't matter because the other copy will make Rh for you, right? So if you look at the red cells of the person, there will still be Rh on the surface because of that copy, right? It's like the cookie cutters I was telling you about. To make star-shaped cookies, you can have two cookie cutters or one cookie cutter, right? You will still be able to make the shape, right? And having both, sure, you can make it faster, but it's like this is not about making it faster or more. It's about whether you can make it or you cannot. If you can make it and you can put it on the red cell, then you will be Rh positive. Okay, so might as well draw the last case here. For someone who is negative and negative, if they inherited the negative allele for Rh, then it's like they don't have the gene to make the Rh. They don't have the cookie cutters to make the cookies. So no Rh on the surface, and this person would be Rh negative. Is that a little bit better, or you have a follow-up question? Negative does not mean absence. Absence what? Absence allele is just a name. Yeah, so if someone is Rh negative, okay, let's write it down. What does Rh negative means? Rh negative means no Rh antigen. Antigen is just a protein, okay? No Rh antigens on RBC. Right? Rh positive means Rh antigens are present on RBC. Okay. Does that answer your question, or anyone else have follow-up questions? You got to understand this RH business before we go deeper in it. The whole note, the whole 45 minutes of notes is about the RH. Okay, anyone else? Just ask if you have a question. Like I said, someone else probably is thinking the same thing. There are no bad questions. Okay, let's practice drawing some of these things, okay? Draw RBC for each blood type. Maybe after you draw it out, it will make more sense. Let's say we want to draw someone who's A positive. I'll do this one with you. A positive. There is your red cell. If a person is A positive, what would be on the surface of the red cell? Well, A means there will be A, right? And what does positive mean? What what else is going to be on it? That's right, RH. That's an A positive person. Okay. But like, in reality, it's not just a single A and a single RH, right? Like A is going to be like all over the place, right? Like all over the place, and you're going to have RH all over the place, right? But, of course, for simplicity, we're going to do it like this. Do you understand? If you do, please draw AB negative. And I'm going to give you 30 seconds to do that. There's your red cell. AB negative. What's going to be on the surface? All right, anybody want to share their answer? What's going to be the AB negative? Okay, thank you, Vivian. Anyone else want to give it a try? Thank you, Cheyenne. Okay, so AB, right? AB means there will be A, and then B means there will be B. And this person is negative. If they're negative, should we put the RH on or no? What does negative mean? Negative means no RH antigens on the RBC, so that's it. Okay, that is it. Okay, let's try another one. O positive. O positive. What's on the surface? Okay, please let me know your answer. You don't have to message to everyone. You can just private message me because I need to know if you get this. Okay. Okay, good. Getting a few answers coming in. Anyone else? Yeah, so in this case, the person is type O. You do not put O on it, okay? There's no such thing as O, right? O is the absence of A and B. So no A, no B, but the person is positive. So we put the RH on. Okay? You can put one RH, two RH, doesn't matter. You don't need to put little I on, okay? The little I is the gene, right? It's the allele, right? Sure, this person is little I, but that's just the gene. The gene's not going to be on the surface of the cell, right? Okay. The surface of the cell is the expression of those genes. So little I means you cannot make A, you cannot make B. So it's just RH and RH. Any questions before we go deeper into this thing? It's gonna get more complicated. I'm telling you. So ask away. We're gonna skip the break today, by the way. I'm gonna go all the way to 10:30 and then we're gonna end the class, okay? So that you have some time to do the quiz, right? For today, just so you know. Okay, well, that's the um that's the genetic makeup and the expression of the RH. Let's take a look at um blood transfusion. Okay, blood transfusion. So um let's just there's a question. What time is the quiz due? Yeah, the quiz is due midnight tonight. Okay, so just make sure you get it done before the end of the day. You should be fine. All right. Okay, so let's talk more about blood transfusion. So let's think about this again in the context of ABO to kind of help you, ease into the RH blood transfusion. Right? I know we talked about this already at the beginning, but just, humor me, stay with me. Uh, a person with type B blood has what kind of antibodies? Has anti-blank antibodies. What's the answer? Yeah, you make what you don't have, right? So a person type B blood will have anti-A antibodies. Similarly, a person with type A blood has anti-B antibodies, right? And if they are type O, then they have anti-A and anti-B antibodies. Okay, so hopefully you understand that, right? Now, for all intents and purposes, for like our purpose, these people pretty much are born with these antibodies. Okay. If you're a type B person, you are born with anti-A antibodies. If you're a type A person, you are born with anti-B antibodies, right? But that's not the case for RH. Okay. So let me just make a quick note here. These antibodies are already there when the person is born. Okay. Again, not straightly true, but close enough. Right? For our purpose, we would just say it's already there when they were born. Okay. However, this is not the case for RH blood group. Okay, I'm going to try to explain this to you. So, after you copy this, I want you to think about the following question. Who's going to be producing anti-RH antibodies? Are the positive people going to be making anti-RH antibodies, or are the negative people going to be making anti-RH antibodies? I'm going to do a poll to see what you guys think. Okay, who's going to be making the anti-RH antibodies? The positive people, negative people, or both? Who makes anti-RH antibodies? That's the question. Okay, anyone else? Just choose something and then we're gonna move on. Okay. Most of you have the right idea. Uh, the correct answer is negative people will make antibodies for RH. I will tell you why. So first of all, let's say this is a RH positive person. RH positive person, if you draw it out, we are just focusing on the RH right now, okay? Forget about ABO. If we just focus on the RH, if a person is RH positive, then there will be RH on the surface. Then there will be no antibodies, no anti-RH antibodies. Remember, you cannot make antibodies that would target things that you have. If you make anti-RH antibodies and you are RH positive, those antibodies will attack your own cells. And that's not good, right? So positive people will not make anti-RH antibodies. Okay. RH negative people, on the other hand, they do not have the RH antigens on their surface, and therefore RH is foreign to them. It's not something they have in their body. And as a general rule, you will make antibodies to attack things that you don't have. So they will have anti-RH antibodies. Okay, just like over here. If you are type B, you are not going to make anti-B antibodies because it will attack yourself. So you make anti-A. You make things, you make antibodies for things that you don't have. So that's why only negative people will make anti-RH antibodies. Okay, for those of you who chose the wrong answer, do you understand now? Follow-up questions? Okay. Now here is the thing I'm trying to tell you, right? And I have, I'm not sure if I am making this clear. For the ABO blood group stuff, I told you the antibodies are pretty much there when the person is born. But that's not the case for RH. Okay. I'll put a note here, and this is very important note. Okay. A RH negative person is not born with anti-RH antibodies. It's the same thing. A baby is not born with, listen carefully, a baby is not born with anti-chickenpox antibodies. They're not born with anti-COVID antibodies. Tell me, how does a person get anti-chickenpox antibodies? Okay, yeah, some people say vaccine, yeah, from his mom. Okay, yeah, let's not talk about that yet. We'll talk about that later. But like for most of us, if we want antibodies for a disease, um, what needs to happen first before your body can make antibodies to attack them, right? Vaccines, sure. What else? That's why you have to get sick, right? You have to catch it. You have to catch the chickenpox. You have to catch the COVID. You have to catch the flu, right? Okay. Getting a vaccine can kind of jumpstart that process. We'll learn about it in lecture 3. But the point is this, you're not gonna be able to make those antibodies until you are exposed to the thing you're trying to create the antibodies for, right? Okay? And that's the case with RH. An Rh negative person is not born with anti-Rh antibodies. The person needs to be sensitized. That's the word I want you to learn. Sensitize. Needs to be sensitized first before they start to make the antibodies. Okay? And sensitized means, yeah, exposed to positive blood. Okay, so there are different ways of sensitizations. There are two ways of sensitization. Probably there are more, but I'm just gonna teach you two, okay? Two ways of sensitization. Number one, okay, just think about this. Like don't go crazy and think about obscure things. Just gut feeling, straightforward answers. If you have negative blood, what's one way that you will be exposed to positive blood? Why or how would positive blood get into your body? That's right, transfusion. Okay. Rh negative person transfused with Rh positive blood. It can happen. Okay. Mistakes, sure, but sometimes on purpose. If a male goes into the hospital and he's suffering from severe blood loss, we don't have time to check his blood type and everything. By default, you give him O positive blood. Okay, I'll tell you why that is okay. I mean, like why they do that in a second. Okay, so if the male happens to be negative, then now they are exposed to positive blood and they are sensitized. Okay. What's another reason? This is it's only gonna happen in female in women. Okay. What situation will a negative Rh negative woman be exposed to positive blood? Pregnancy. That's right. Okay. Pregnancy. Rh negative mother carrying a Rh positive baby. Fetus. We'll say fetus. Okay. We're going to talk more about that in a bit. Is this okay? Give me a sign that you understand. Yep. All right. So here we go. Here we go. Okay. Let's talk about, we're trying to talk about blood transfusion, right? So generally speaking, generally, we want to prevent sensitization. We want to prevent sensitization. Right? So Rh negative people can donate to other negative or other positive. Remember, if you are Rh negative, that means there's nothing on the cell. And when you give it to other people, there is nothing to react against, right? So negative blood, you can give to both negative people and positive people. If you're Rh positive, you can only give to other positive people. Okay? But then there is a catch. There's a catch. There's like always a catch. I'm running out of colors here. Okay, here's catch. A Rh negative person can receive Rh positive blood once. Okay. Why is that? Well, if I'm negative and I have never been exposed to positive blood before, then I do not have the anti-Rh antibodies in me. So when the positive blood goes into me, I will not have the antibodies to attack the cells and I will accept the cells. I will accept the red cells, and that's fine. However, moving forward, my body will start to make antibodies because now I'm exposed. Now I am sensitized. So I will have Rh antibodies in my system. So if I receive Rh positive blood again in the future, then it's not good because I already have the antibodies produced from the first exposure, and now they will attack the incoming cells. So you have like one free chance, so to speak, if you are negative. All right? But generally, we don't want to use up that free chance unless it's like, emergency situation. For all the questions you will be doing on the test, whenever we are doing blood transfusion and I ask you if it's okay, if it's safe or not safe, you have to assume you are trying to prevent sensitization. So even though it's okay for negative people to receive positive blood once, you don't want to do that uh, in the questions. Okay, we assume we're trying to prevent sensitization always. Having said that, let's take a look at some examples. And again, those videos I was telling you about in the lab folders, I went through a few of these examples with visual aids to help you understand. Please watch them if you are very confused by this. Okay, so let's do some examples. Can you give a negative blood to AB positive? Okay, A negative to AB positive. We have to look at the two blood groups separately. Okay, you look at the ABO first, and then you look at the RH. So can A give to AB? Is that okay? A to AB? A to AB is okay. If you remember the cheat sheet I gave you last time. Okay. Look, A to AB is fine because in AB there are no antibodies, right? So the A red cells will be accepted. And then you look at the RH, can negative give to positive? Yes, negative can always give to positive. Right? So both blood groups are compatible. This is safe. Let's do another one. What about O positive to A negative? O positive to A negative. Give that a try, and then I will take it up in a minute. I just need to look for something else in the study guide. All right, what do you guys think? Safe or not safe? Safe or not safe? Well, O to A. Take a look here. O to A, that's safe, no problem. And then this is the RH transfusion chart. Positive to negative, not safe, right? Okay. Even though they can receive it once, but like I said, we want to avoid that. So if one of them is not safe, then it's not safe. Okay. Every blood type needs to be compatible before you can do the transfusion. Okay. So why don't we try this question here? This is number 8 in the study guide. Oh, that looks terrible. Let me try to copy it again. There you go. I'm gonna give you a couple minutes. Okay, 3 minutes to do this. Okay. These are the donors. These are the recipients. Like, can A plus give to AB negative, right? So take a moment, fill it in, and I'll take it up with you. Okay, hopefully you have a chance to try this out. I think only a handful of these things are safe. Let me see. Okay, this is safe, 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 safe. That's it. Everything else is not safe. Everything else is no good. Is there a particular one you want me to explain? Any one of these not make sense to you, let me know, and I will elaborate. So last class, I was telling you, right? A lot of people call type O blood universal donor, but as you can see here, that's not really the case, right? Sure, O is universal within the ABO blood group, but when we do blood transfusion, we can't just look at ABO alone. We have to look at, RH as well. And so this person being O positive, you actually cannot give it to A negative. So O is not universal, um, all the time. You need to consider other blood group as well. And I also told you, besides ABO and RH, there are other blood groups, right? And those needs to be taken into consideration as well. There are 40 different blood groups that I showed you, many of which we are not going to talk about, but they still play an important role when it comes to blood compatibility. So we are good with the transfusion. Everybody got this right? No questions, no concerns. Okay. Let's just do another question here from the study guide. All right, there we go. This is number 7. I'm just gonna change this to B negative. Okay, just like mix it up a little bit. So if you are A positive, who can you receive RBC from? Well, if you're A, you can receive from other A's and you can receive from other O's. And if you're positive, you can receive from positive people as well as negative people. And so these are all compatible donors for you. Who can you donate the blood to? A can donate to other A's. And if you follow the donation table here, let me just move it down a little bit. You follow the donation table here, you can see A can donate to AB as well. And since they are positive, they can only donate to other positive people because otherwise they will sensitize the negative person, and we don't want that. If you are B negative, who can you receive from? I'll give you a minute to think about that. Who can B negative receive RBC from? That's right. Other B negatives and O negatives, right? They cannot receive from positive people. Who can they donate to? They can donate to B negative, B positive, AB negative, and AB positive. Just follow the pathway, right? And you will be able to figure it out. All right, that's it for blood transfusion. Um, there are other things we need to talk about. I'm just deciding what to do next, so that makes more sense. Let's talk about the sensitization more. Okay. and then we're gonna do some other questions. Just quickly recap. Sensitized means a negative person is exposed to positive blood. Only negative people can get sensitized. Okay? If you're positive, you're not gonna get sensitized. Okay? It's like you already have positive blood. You cannot get exposed to it. And here is something I want to emphasize. Okay? In the past, student gets confused with this. When a person is sensitized, it does not mean their blood type changes. Okay? It's not like if you're negative and you get exposed to positive blood, then you become positive. It's not like that. Right? Sensitized just means you are negative and you get exposed to positive blood by one of these methods, and now your body will make anti-Rh antibodies. Okay? So I'm going to give you a note that explains sensitization through pregnancy. Right? Sensitization through pregnancy. Rh sensitization, because you can get sensitized by other blood group as well. Okay, so but we're focusing on Rh sensitization through pregnancy. There we go. So we will consider an Rh negative mother and an Rh positive father. What's the genotype of Rh negative mother? What's the genotype for the Rh negative mother? That's it. Negative, negative, right? There's only one way for her to be negative. For the father, though, he could be positive, positive, or positive, negative. To make things easier, we're just gonna assume he's positive, positive. Okay? I'm gonna make a side note in case you look at this later and you don't understand. Okay? And positive, negative, also possible. Okay? But we will use positive, positive to make it simpler. Okay, you don't have to write this down. It's gonna be in the notes when I post it. Okay, so let's do a Punnett square and see. I mean, you don't really need to, hopefully, to understand this, but why not? The mother is negative, negative, so all her eggs will be negative. Father is positive, positive. All the eggs will be positive. And if we do the Punnett square, you will see there's 100% chance, it's guarantee the child and all the children will be Rh positive, right? It's guaranteed. First kid's going to be positive. Second kid, if it's the same father, it's going to be positive. And all the children, all the kids, right? It's going to be positive. So let's take a look at the first pregnancy. First pregnancy. Okay, I'll try my best here. This is the mother's blood. Mother's blood. And mother is Rh negative. Okay, so let's draw a bunch of her red cells. There we go. Right, these are all her red blood cells. Happy faces without the eyes, basically. And she's negative. I'm not talking about ABO, so we're not gonna draw the A's and the B's, but she's negative, so I'm not gonna put any RH on them. Right? These are RH negative RBC. Nothing too complicated. And we're gonna draw the baby's blood. Baby's blood right here. There we go. Baby. Baby's blood. And because the father is Rh positive, and we look at the Punnett square, the kid will be Rh positive. Okay. He's positive slash negative, which means he's positive. So the baby's blood, the fetus's blood, if you would, and we're going to have these red cells. Okay. And on the surface of the red cells, we will have RH. The baby will have RH. There you go. So these are RH positive RBC in the baby. So what prevents the mother's blood and the baby's blood from mixing with each other? What is the thing that separates the mother and the baby? Anybody? Starts with a P. That's right, the placenta. Right, separates the mother and baby, the mothers and the babies, I guess. Mothers and babies' blood. All right. So there's no mixing going on. So far, so good. All right. So here's what happens. If the placenta is damaged during pregnancy or during labor, like as in like giving birth, then the baby's blood might enter the mother and she will be sensitized. Okay? She'll be sensitized. Right? So if we damage the placenta, yeah, I'm not gonna erase this thing. It's hard to draw it, I guess. But here's the thing, okay? You decide if you want to draw this or not. If we damage the placenta during labor, or sometimes it could happen during pregnancy, these fetal cells, they will now move into the mother's bloodstream. And she's now exposed. Okay? She's now exposed. She's now sensitized. Okay? Now, suppose it happens during labor. Okay, if it happens during labor, is this going to be a problem for the baby? Why not? Elvis says no. Sheila said no. Why not? The mother will now produce Rh antibodies. But by the time antibodies are made, the baby, where's the baby? Would be delivered already. Okay. You don't make antibodies like right away. Right? It takes time. That's why if you're sick with the flu or something, it takes a couple of days before you start feeling better. That couple of days is basically how long it takes to make the antibodies. Right? You receive a vaccine, you're not protected like immediately. You can still get sick while your body ramps up the antibody production, right? So it takes some time for the antibodies to be made, and that's the situation here, right? Even though the mother is sensitized, by the time she makes the antibody, baby's going to be delivered, and it's going to be fine. Okay. Now, if it happens during pregnancy, like early on in the pregnancy, then we're going to have some complications, but we're not going to focus on that at the moment. Any questions about the first pregnancy before I tell you about the second pregnancy? Okay, here we go. Second pregnancy. Second pregnancy. So let's draw this again. Mother's blood. And she's negative, right? There are the cells. And how is it going to be different this time compared to before? What will be in her blood that wasn't there the first time? Okay, this was the first pregnancy, now second pregnancy. Because she was sensitized in her first pregnancy, the baby's blood got into her system, she will now have anti-Rh antibodies. Okay, so these are anti-Rh antibodies. Okay. She made it because she was exposed to positive blood. And these antibodies stays in your system for quite some time. And so we're gonna have the baby's blood now. The baby's blood. Sorry, I gotta restart again. Sometimes like if I use these straight lines and auto shaves, the program gets all weird. And there we go. This is the baby's blood. And he's also positive, right? Because of the father. Let's try it to all the baby's blood. There we go. I remember they are 100% guaranteed to be positive because of the parents' genetic makeup. Okay, you still have the placenta here. And this is what's going to happen. The antibodies that are produced in the mother's body will cross the placenta and it will cross-react with the baby's blood. Anti-Rh antibodies cross the placenta and basically attack the baby's Rh positive RBC. This results in, there's a name for this kind of condition, results in, let me choose, I don't know, orange, hemolytic disease of the newborn, or HDN for short. Okay, I'll explain more, just copy first. You know what? I think we're making good time. So, why don't we take 5 just for you to catch up and rest a little bit. We'll resume at 9:45, okay? 9:45. If you have any questions, leave it in the chat. Otherwise, just copy whatever you need to copy. Okay, so I want to go back to something I said earlier, right? Here, the mother does not have the antibodies because she was not previously sensitized. Okay, so here's the situation. And I know this because I told you my wife, works in the blood department at uh, at the hospital. So she said if a woman comes in and she's under the age of 65, for example, I think 65 is the cutoff for their policies, and she needs blood urgent, then you would give them O negative. Okay, but if it's 65 and above, then you give them O positive. If we're talking about like emergency transfusion, if it's a man coming in, O positive. Okay, of course, like if you have time, you would check their blood type and everything, right? But why does this work? Well, if the person is under age 65, then they are considered like reproductively active. And so you don't want to give them O positive blood because that could potentially sensitize them if they're negative, and that's going to cause problem in their first pregnancy if their baby is positive, right? 65 and above, not reproductive age, generally speaking. So it doesn't matter. Even if you sensitize them, it's fine. Okay. We worry about later if they need a second transfusion. At least we're gonna, be able to help them first, right? Men not gonna have babies. That's why it doesn't matter. If you sensitize them, you sensitize them. That's fine, right? Like it's not gonna have this Rh incompatibility, but they will make a knock in the system, and if they come back again, then we have to know not to give them positive blood again in the future, because now they would have the antibodies in them, right? We good? All right, I hope so. It's a lot of stuff. It's a lot of stuff. I am fully aware of it. And this is the professional drawing. It's the exact same thing I was trying to convey to you. Um, so, mother is negative. That's why there are no spikes on the red cells. And then these little triangle are Rh positive. And then there are like hemorrhaging at delivery, bleeding, damages the placenta. Then the fetal blood is going to go into the mother's blood. Mother will now produce antibodies, but it's okay because the baby is out. But in future pregnancy, it's not okay because the antibodies will cross the placenta, attack the baby's cell, and they would undergo hemolysis, bursting, right? So that will cause hemolytic disease of the newborn. There's a fancy name for it, erythroblastosis fetalis. Okay, I'm just putting it there in case you run into it when you read some of this stuff online. Now, the symptom severity varies. It depends on the blood group that we're talking about. Okay, Rh is not the only blood group that can cause hemolytic disease of the newborn. Remember, there are other blood groups, right? Like Cal is another one that could cause quite severe HDN, hemolytic disease of newborn. Okay. ABO incompatibility can also cause problems, but not as severe. There's a question. So can the mother check if they have antibodies before getting pregnant again? Okay, we're going to get to that in a second. Now, what are some of the symptoms? Well, the symptoms are going to be in the babies. The mother's going to be fine. Okay, it's the baby that's having the problem. The baby will have anemia, which is expected because a lot of the red cells are dying, right? Jaundice, because I think last week I told you when red cells die, the hemoglobin gets metabolized into this thing called bilirubin. And normally we pee it out and we poop it out, and that gives the yellowish color. But when there's so much of it, they start to build up in the body, and it gives this yellow hue skin tone to the person, and that's called jaundice. Enlarged liver and spleen because these organs are working extra hard to cope with the red cells dying. Remember, that's where red cells go when they are broken down, right? To the liver and the spleen. Severe edema, that's swelling. Because the red cells are dying, the osmotic pressure will reduce quite a bit, and you're not able to retain the fluid inside the blood vessels, and they, leaked out from the vessels, causing swelling in the tissue. Okay, and like I said, the symptoms is gonna vary in severity, and the disease itself is gonna vary in severity, and the treatment is going to vary as well. Okay. These are my daughters. This is the older one, I think. This is the younger. I can't tell anymore. But they were both born with jaundice. Okay. Um, I am AB. I'm AB positive, and my wife is O positive. Okay. And both of them are B positive. B positive. Okay. So RH, in terms of RH, they're fine. Okay. RH, there's no incompatibility. But my wife is O positive, which means she has anti-A and anti-B in her bloodstream, right? So these anti-A, anti-B antibodies can actually cross the placenta and attack the baby's uh, B blood, okay? The anti-B specifically, okay? Anti-A is not gonna do anything, but anti-B will attack these B cells, type B cells in the baby's body, and it causes a little bit of hemolysis. It's not very severe, but severe enough for them to be jaundice. And because it's not very severe, one of the ways to fix the jaundice is to put them in this chamber. Okay, the UV light is what's giving the blue color, and UV light is supposed to help break down the bilirubin in their body, right? In the past, you probably have heard of this, right? People say just leave the baby in the sun, right? And the yellow hue will go away. It's the same principle, okay? The sun has UV light, and putting them in this chamber is just more like more control environment, right? And it's actually 37 degrees in there, so it's really warm. That's why they don't have to wear anything. And judging from the position, they're fully enjoying it. And this BD light treatment is supposed to help get rid of the jaundice. And you also have to feed them more because it will help them pee more, and then it's going to help flush out the bilirubin. So my first daughter, jaundice, we in Canada they really advocate for like breastfeeding, and so and I'm sure some of you know if you have kids, like if you breastfeed your baby, sometimes the milk is just not there yet, right? At the first couple of days, your body is just not like ramping up the production yet. And so, the babe, my first daughter was kind of dehydrated, because not getting enough milk from the mother, and she was really jaundiced. The light was helping, but then because she's not flushing it out of her system, it's not really going away. So I was concerned and called my mom, and then my mom was like, just feed her formulas, right? It's fine. All of you grew up on formulas and you guys turn out okay, right? You just really need to flush it. And that's what we did, right? So we switched to formulas right away, and then she's ate a lot, and she was able to pee out the bilirubin, and the jaundice went away a couple of days, right? So came the second baby. We were like, well, we're ready for this. Okay. We are experienced. We bought a whole pack of formulas to the hospital with us. And we were ready. Okay. But the second baby, she refused to came out. And again, some of you might know this. If the baby doesn't want to come out, you have to vacuum them out. Okay, there's a little device that they put on their head, and then they pull them out. It's kind of funny because when I told my younger daughter that we had to vacuum you out, she was like horrified. She's like, the vacuum? Like she's thinking about like the one you used to vacuum the house with, and she was really confused. Anyway, so they vacuumed her out, and when you do that, it leaves behind like a really big bruised area in the head, and a bruise is basically damaged red cells. And when you have the damaged red cells, it further elevates the bilirubin, right? So she was really jaundiced, the second one. Okay, even after spending in the Billy lights, and then we feed her the formulas, like she was there for like a good 2, 3 days, as opposed to just one day here for my other one, and she, it just wouldn't go away. Because of the elevated trauma, right, from vacuuming. Anyways, they're both fine, and this is very mild compared to RH sensitization. In these situations, they might need to do transfusion in some cases, and to compensate for the loss of red cells in them. Okay, so there you have it, hemolytic disease of the newborn. Now, can you prevent this thing? Well, ABO blood group, they usually don't do anything about it. It's just very mild compared to something like RH. Uh, but there is a way to prevent sensitization in RH blood group, and that's called RhoGAM. Where is, oh, right here. Okay, RhoGAM. So RhoGAM is artificial anti-RH antibodies. Right? And the purpose of RhoGAM is to prevent sensitization. It does not reverse it. Okay. If it already happened, then RhoGAM's not going to do anything for you. Okay. It doesn't cure it or anything like that. It prevents it from happening. Okay. So what happens is if the mother is RH negative, they will be recommended to take the RhoGAM. Okay? At 28 weeks of pregnancy, as well as receiving the RhoGAM after childbirth, after delivery. Okay? So what does it do? This RhoGAM is anti-RH antibodies that will remove any baby's cells in the mother's body. So let's go back to our picture here, right? If the placenta is damaged and the baby cells goes into the mother, don't draw this, just watch. The RhoGAM that we inject into the mother, these artificial antibodies, will basically attack the incoming cells and destroy all of them before they trigger her immune system. Okay? Before the positive cells trigger her immune system. And that way she remains unexposed, right? It clears all the baby cells in the mother before it activates her immune system. So that's how you prevent sensitization. Now you might be thinking, how come the RhoGAM is not going to, cross the placenta and attack the baby? Well, these are artificial antibodies, which means they are made in the lab, and they actually make it really big. Right? These antibodies are so big that they are not going to be able to fit through the placenta And if you inject it into the mother, it will stay within the mother only. Right? It's not going to be able to get into the baby's blood, and therefore the baby will remain unaffected. Okay? And any antibodies that you inject into the mother, any RhoGAM that you inject into the mother is going to be temporary. Now, it's not going to stay there forever. Right? So next time, if she becomes pregnant again, she would need another round of shots. So that's how you prevent sensitization. Questions? No questions. All right. Uh, let's do some polls first, and then we'll go from there. Oh, actually, there's another thing I need to go through with you. Oh, there's so many things I need to go through with you. Uh, Let me think about what the right order is. Okay, let's pause for a second and talk about some other some other things first, and then we'll go on there. All right, so you're gonna get some of these on the on the test. Here we go. We have some mother and fathers here who misplaced their babies, I guess, at the hospital. And we're gonna use blood types to try to figure out which baby belongs to which parents. Okay? For questions like these on the test, there is no need to do Punnett square. Okay? You just have to remember the following four things. Okay? Four things to remember in addition to, a million other things you have to remember. Four things to remember for these type of questions. Okay. Number one, A, B person, A, B person cannot have what kind of babies? Okay. If you are type AB, it doesn't matter who you marry. What kind of blood type will you not be able to create for your kids? That's right. Cannot have type O kids. Okay. Because in order to have a type O baby, you got to pass on a little I. And if you're AB, you can't do that. Okay. You don't have a little I if you're AB. I need a few more statements. Anyone else want to try? Here, I'll help you start. What about a old person? Huh? What kind of kids can they not have? Oh, Elphys has another one. That's a good one. I'll put it on. Okay, but let's do this first. I guess an old person cannot have type AB kids. Okay. This is regardless of who you marry. All right? Biological kids I'm talking about, obviously. So like if you're type O, you can only pass on little I. And for someone to be type AB, they need to receive IA from one parent and IB from the other. Okay. Elvis is another one. Two RH negative people can only have RH negative kids. Right? The opposite is not true. Okay. Positive people can have negative kids. For example, if they're positive negative like that, then there's a 25% chance they will be negative. Yeah. So positive is not, you cannot write the same thing for positive people. Can I have one more, please? All right, one more statement regarding type O blood. Anyone? No? Nothing? Two old people can only have what kind of kids? Okay. If the parents are both type O, then they both have little eyes only. They can only pass on little eyes and they can only have O kids. Okay. So remember these four things and it will save you a lot of time on the test. So let's see how we can use this. All right. So these are the parents. These are the potential babies. You don't have to start with the first one, right? In fact, don't. Okay, start with something with O or AB in it. Right? Like the second one is a good one to start with. O negative, O negative. According to rule number 3 and 4, if they are both O, then they can only have type O babies. If they're both negative, they can only have negative babies. So baby L is theirs. Right? So they can take baby L home. Rest assured, it's theirs. All right, let's take a look at the next couple. Okay, listen carefully, huh? AB, the mother is AB. That means she cannot have type O baby. So this is not theirs. Okay. Doesn't matter the father is type O. If the mother is AB, she cannot have a type O child. Similarly, the father is type O, so he cannot have AB baby. So that's not theirs. Okay. Baby L is already taken, so the only one that's possible from this list of three, it will be baby H. Okay, so baby H is gone. All right, couple number 4. Mother is O. That means she cannot have AB babies. Okay, so J is not theirs. That means I is theirs. And finally, there's only one baby left, which goes right there. That's how you do it. Now, you just logic your way through it. You don't have to do Punnett square. You just use these four things to help you guide your, you know, answers. Is that okay? There are more practice in the study guide. Help yourself with them. All right, next we're gonna do another thing here. Another back to the PowerPoint. Okay. We're gonna do blood typing. All right. And again, I know I said this already. There are videos on the course shell where me and this other uh, prof, Temjit, we actually did the experiment to show you what it really looks like. So watch it, and it will help you understand more. Okay? But this is how it goes. Okay? We have a blood sample here, and then we read the sample like by row. Okay. This is like it's like a plastic plate, okay, and the plastic plate basically has like wells on in it. So in the wells, we put different antibodies in it. Okay? So for example, in this first well, there are anti-A antibodies in them. In the second well, we have anti-B antibodies. Anti-D is same as anti-Rh, okay? I'm not gonna explain the differences. They are both the same thing. All right. I am not saying the blood has anti-A in it. I'm not saying the blood has anti-B. Okay, that's where the confusion comes from for a lot of students. The wells already have these antibodies in them. Okay, you add the anti-A and anti-B and then anti-Rh. And then you're gonna take the blood sample and you're gonna put the sample into the wells and mix it with the antibodies. And you want to see if the blood reacts with the antibodies. Right? So how do you know if it reacts or not? Well, you're looking for these clumpings, right? There is a picture earlier here. If there are no clumping, then there are no reactions. And then if you see the little flicky stuff, then there is a reaction, okay? So over here, because the blood sample reacted with anti-A antibodies, you can see the clumping here, the agglutination, then I know the person has type A blood. Okay? It has A on the red blood cells because it reacted with the anti-A. No reaction with the B antibodies, so the blood does not have B antigens. And it reacted with the RH, so this person is A positive. If it reacts with the antibody, then it must have the antigens on them. All right, let's take a look at the next one. Antibodies already in the wells. We're adding the blood to it. No reaction, no A. Reaction with antibody B, the person must have B antigens on them. And reaction with RH as well. So positive. Please try the next two. If you have questions, please ask. All right, what's the next one? Okay, so this one, reaction with A because of the compound, that means it must have A on the red cells. There are also reactions with B. So this person is AB. And also reaction with RH. So RH positive. This person is AB positive. Last one. No reaction with A or B, then it must be O. And also no reaction with RH, must be negative. So that's the blood typing. Any questions? Okay, there are more practice in the study guide. I hope you guys are working through them, right? Look, more practice, uh, and then check the answers when you're done. All right, let's do some polls. Go ahead. Okay, anyone else? All right. I'll show you guys the results. The correct answer is D. Okay, Rh negative mother who is carrying a positive fetus. It's not A. If the mother is positive and the fetus is positive, first of all, the mother will not be sensitized. Only negative people will get sensitized, right? Right? That's the first thing to know. Number two, you cannot give RhoGAM to the positive mother because RhoGAM attack positive red blood cells. If you give the RhoGAM to the mother, then now the mother's blood will be destroyed. Right? She would experience hemolysis. Right? It's not safe to give RhoGAM to positive people. Okay? You're giving the RhoGAM to negative mother who carries a positive baby in case the baby's blood goes into the mother. Then the RhoGAM will clear it out of the mother system before she gets sensitized. That's the purpose of RhoGAM. Another question here. Uh, at first glance, there are two correct answers, but read it carefully. Only one of them is correct. Okay. Very good. Most of you got the right answer. It is C. If the mother is negative and the father is positive and the baby is positive, then we potentially could have one of these situations, right? B is not the right answer. No, nobody chose it, which is fantastic. But the mother is negative and the father is negative. It's not possible for them to have a positive baby. So this situation is not even going to happen. So that's not it. Okay, and positive mother is not going to get into this problem. So those are not the answers either. Okay, matching with the babies. Matching with the babies. Okay, take a moment and try this out. Okay, let's take this up and see. Again, use the four rules that I have given you and go from there. Let me just copy this to the OneNote right here. All right. So, uh, sometimes you just have to start working on it and see whether it works or not. So first couple, the mother is AB. That means is not this baby and is not that baby. AB cannot have O, right? And the mother and the father are both negative, so it's not possible to have a positive baby. So the correct answer is L for the first couple. So L is gone. Next one. I'm gonna work with something with a type O in it. Okay? It's probably easier to eliminate choices with a type O parent or type AB parent. So the father is O. That means he cannot have an AB baby. So that's not it. Both of them are negative. They cannot have a positive baby. So M is their kid. All right, let's see here. This one you have to think a little bit more, I guess. Both of the parents are type B, so they do not have an IA allele to contribute, and therefore they cannot have an AB baby. It must be K. And if you're wondering, how can two type B person produce a type O baby? Well, they both must be heterozygous. IB little I, right? And there's a 25% chance they will be able to produce a type O baby. And so the answer is LJKM. All right. So practice with these things. It's not going to get harder than this. There will be matching questions like this kind on the test. All right. Another question here. Go ahead. Okay. I have people selecting everything, even an F. There's no F here. I guess that means not sure. Um, Anything else before I take it up? Only one of these are safe. On the quiz or test, if they say select all correct answers, then there are more than one, right? I'm just gonna take it up then. Over here, first one, A positive to B positive is not safe because A cannot give to B. B has anti-A in them. Next one, O can give to A, but positive cannot give to negative. Not safe. AB cannot give to B. The B has anti-A, which will attack the incoming AB. A can give to AB. Negative can give to negative. That's the safe one. And last one is not safe because positive cannot give to negative. Okay. Two more. Two more. You do not need to do the Punnett square. Just need to use the logic that I gave you. Okay. Keep trying if you want. Otherwise, let's take this up. So AB cannot have O. I keep on telling you that, right? So these are not possible blood type for the kids. Type O cannot have AB, so that's not possible. And that's it, right? You literally can do this question in under 15 seconds if you apply those four rules that I gave you, right? And of course, time is important on the test. You don't want to, waste too much time on something that can be done easily. You don't want to be drawing Punnett square when you don't have to. Last question, and then we're done for the day. All right. If a person is type A, they will make anti-B antibodies. And if they're negative, they will make anti-RH antibodies. So the only answer that matches that would be B right there. Okay, that's it for today. We'll do the rest on Wednesday. Please be sure to do the quiz. You would just go to the quiz here, and then you would do it. Okay, make sure you have the what do you call that thing? The lockdown browser. Uh, if you haven't done the practice quiz, which is right here, study resources, self quiz. I don't think a lot of you have done it, okay? Do that first. It will help you do better on the actual quiz. Don't just cold turkey it, okay? Like is the quiz open? I think so. Why is it not? See it right here, right? It should be. Let me see if I go to student view. Okay, so this is what you see. You click on the folder and then it's here. Yeah, you just click on that and then, it will tell you blah, 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 40 minutes, one attempt. You gotta download the Respondus lockdown browser if you lost it somehow, and then you start, and that's it. Okay, you have until midnight to do it. That's it. Okay, thank you guys. Good luck. See you on Wednesday. Bye for now.